Hi, this is Dr. Deepak Modi. I am from the ICMR National Institute for Research in Reproductive and Child Health, and I'm a scientist there. So I chose to do biological sciences as a profession because I could associate with how human bodies function, how animal bodies function, and what things go wrong there to cause diseases. And this prompted me to do my bachelor's in zoology, which was followed by a master's in human physiology and then a PhD in human genetics. During this period, I met some fantastic mentors who trained me how to ask research questions, how to answer them, and then eventually I got a faculty position here at this institute, and that was the beginning of my journey as a scientist. The motivation to do this work is the primary fundamental nature of the question. It allows us to explore how the embryo talks to the endometrium, how the early placenta is formed, and also, it has a social relevance because the mandate of the government India implies reduction in maternal mortality, reduction in neonatal mortality, and pregnancy-related complications. So you're doing very fundamental research. We can answer very basic problems and help the government to solve the issues related with women's health. So addressing the questions of embryo implantation and early placentation have always been challenging because of unavailability of good systems. With the advancement in science and technology, three major technologies are shaping our research currently. One is our ability to sequence RNA at single cell resolution. Second is ability to grow these systems now in 3 dimensions in form of organoids and organ-on-chip. And number 3, our ability to image the system at high resolution in 3 dimension and also with time. These 3 technologies combined together are now helping us to address the problem of how embryo implants and how the placenta is formed. We must understand that embryo development, its implantation to the mother's womb and forming the placenta is a continuum. It's not a one-time process. Now, it is neither ethical nor justifiable to collect tissues from humans, especially during pregnancy, and study them in the lab. So obviously then we resort to animal models, but animals are not humans. Animal placenta, the animal endometrium is way different from the humans, and this posed a very significant challenge to our work for a very long time. We could only study these processes in the animals and probably do some 2D cultures of the human cells to answer this problem. But now we have the system where we can grow this endometrium in 3 dimensions. We also now have abilities to make organ-on-chip like a placenta-on-chip, and we can study these interactions in a lab dish and monitor them live. In collaboration with IIT Bombay, Professor Abhijit Majumdar, we have developed this wonderful placenta-on-chip system, which allows us to explore how things from the mother goes to the fetuses and how this crosstalk happens and how things go wrong there to cause pregnancy-related disorders. This combined with imaging in the HCS system, which we have from Revati, allows us to study the fundamental nature of the problem of the embryo interacting with the mother's cells and how it embeds into the endometrium, leading to the formation of early placenta. So this beautiful process can now be monitored in live with the high content screening system, and we just don't monitor in live, but we are now quantifying it at single cell resolution to address these questions. So trust me, these technologies are here to stay. They're not going to go anywhere, and they're just going to get better and better. Enough we have treated animals, especially mouse, as a 25-gram humans. We cannot use the data from the mouse and believe it's true for the humans, and these systems are the ones which possibly are the answers for all the problems that we have not been able to address it here. I understand it's challenging because we are unable to have single medias. Currently we are unable to grow these in for long term, but once these challenges have overcome, this 3D cultures, the organoids and their imaging and quantification is going to transform lives, transform patient care. So be with it, address it right away. I would say innovate, translate and change human lives. Thank you so much for having me here.